Welcome everyone to this week's edition of the Extracellular Vesicle Club. Recently, I've been talking with my son, who has a friend who posted a dance video on YouTube, and he got 38,000 likes within the first few days. So I'm not going to ask Rienk, Johannes, and Carla to do a dance here, but I think nevertheless we're going to have some very exciting fun. And I'd like to just ask everybody, as we talk about our exciting topic today, which is coagulation and extracellular vesicles, please put your comments and your questions in the chat box. And at the end of the presentation, you'll be able to unmute yourselves, show your video if you want to, do a little dance, whatever you want to do, and have a conversation then with our presenters today. But we have some, as I said, very exciting work here that's going to be featured, and not just recent work, but also work that has come together over the last century. So it's really nice to see how this field has developed and how some explanations have come along for phenomena that we knew about a long time ago, but that involve our dear extracellular vesicles. So Rienk Nieuwland, I'd like to introduce you very briefly. You served in many roles with ICEV, but also with some other very important scientific societies, including ISAC and ISTH. And you've really been, we were just talking with Johannes just now about how the topic of coagulation is extremely important in many areas. Sometimes it's not so well represented at ISAC meetings. Maybe today's EV Club will help to change that. And Rienk, thank you for being here. And for all that you've done for the field and for ICEV in particular. So Rienk, I'd like to hand the screen sharing over to you and Carla, and please go ahead when you're ready.
Thanks, Ken, for the introduction. Carla, can you share the
Sure.
screen? Good. So Ken, thanks for the opportunity to present our work. So I made a few introductory slides trying to put the paper into perspective. As Ken said, it has been essentially a work, and Johannes will mainly talk about that. So Carla, if you can manage.
Okay. Sorry.
Yeah. Please go to the next one. So about 1.5 years ago, a paper was published in Nature by an English journalist, Alison Abbott. And this paper was based on a series of interviews he had with people who are working in the research field. And this paper is about the intercellular communication, the role that vesicles may play there. Can you go to the next slide, Carla? So this is a figure from this paper, and I think this is not a surprising figure. This is a sort of figure we see in many papers. The idea is that vesicles can exchange messages between cells. Next one, please. Yeah, thanks. So, of course, it's very appealing that vesicles are involved in intercellular communication, but in all reality, and I've said this many times before, also at ISAC meetings, if I'm thinking about vesicles in human saliva, human urine, and other body fluids, for me, it's very difficult to imagine that all these vesicles are really involved in intercellular communication. Next slide. So, in this Nature paper from Alison, there was one guy stupid enough to make this statement, and I was this guy. And this statement is, I've never seen conclusive evidence that extracellular vesicles do anything in vivo. And this was based on my idea that if I'm looking at vesicles in body fluids like urine and saliva, there really is no evidence what these vesicles are doing. Next slide, please. So now this paper from Johannes and Carla has come out. I was also involved. It has a very long title, but I think that's not the importance here. Can you go to the next one, Carla? Why I think this is an important paper. So this paper was published by the end of 2024, and it already has gained a lot of interest. It has been presented at multiple clinical meetings. There's even a podcast being made. Unfortunately, it's in German, so that's not helpful for most of you. Next slide, please. It has also been picked up by many patient platforms. So also patients are interested in these findings. The next one, Carla. So why I think this paper is important is that I think, for me, this paper is game-changing, because I think this paper gives some insight and provides some evidence that vesicles may really do something in vivo, and they are involved in coagulation and protection. Next one. That's my last one. So I hope that in this presentation, Johannes and Carla will be able to convince you that vesicles really play a role in vivo and that they are involved in coagulation and protection. And I would like to give the floor to Johannes and Carla now. Please.
Thank you, Rienk. So I would like first to say that this talk is mainly about human physiology and not pathology, and is divided into 3 parts. First, I will give a very short introduction on blood coagulation, and then I will show you the studies that have been done in the past decade, and then Johannes will take you back 100 years. This is a very simplified scheme of blood coagulation. Blood coagulation is initiated when tissue factor binds and activates factor 7. The complex tissue factor 7A then activate the serine protease factor 10, and then it starts basically a coagulation cascade, which eventually will turn in the formation of fibrin meshes, so blood clot. So in physiological conditions, tissue factor is not exposed to blood. It's confined on the extravascular compartment of the vessel wall in the adventitia, and only upon vascular and tissue damage it becomes exposed to blood. It binds factor 7 activate factor 7 and induces the formation of clot. Now, tissue factor and factor 7A are not the only players on triggering coagulation. The third player are procoagulant membranes. It is very important, as tissue factor is a transmembrane protein, and it requires, so the coagulation required procoagulant membranes that can be a cell or a vesicle, meaning that in the membrane of this, in this membrane, the phosphatidylserine residues will be exposed to the extracellular environment, and this will result in these, the surface will be negatively charged. In 2011, the group from Rienk investigated the clotting activity of saliva. They showed that by adding saliva to vesicle-depleted plasma induce coagulation. And they have associated this activity to the vesicular component of saliva. They have indeed shown that these vesicles expose tissue factor. And indeed, by adding the vesicle to plasma, they show that these vesicles induce clot formation, and this could be completely inhibited by pre-incubation of this vesicle with anti-factor 7 antibodies. 2 years later, vesicles exposing tissue factors were also isolated from human seminal fluid. And by adding seminal fluid or seminal fluid-derived EVs, they have demonstrated that there is a fast clot formation, and this is completely dependent on tissue factor and factor 7. In 2020, Johannes, Rienk, and Yong Hu, that at the time was a PhD student, investigated the human milk, mother's milk. And in this graph, where you see here plasma clotting time, basically the shorter is the time, so the lower is the bar, the more potent is the trigger. So they showed that milk has indeed clotting activity, and they showed that even when milk is highly diluted, so 2,500 times, it is still able to reduce clot formation in a significant way. the time to clot formation in a significant way. They have separated by centrifugation the pellet and the supernatant components of milk. Of course, in the supernatant is enriched with these vesicles, as shown in this nice cryo-electron microscopy image, where we see phosphatidylserine positive vesicles. And they have also shown that in this pellet, there is tissue factor. What they have also done further is to isolated vesicle fractions by size exclusion chromatography. They have taken the fractions, they have shown that those fractions where vesicles are, they possess tissue factor, they have coagulant activity, and this coagulant activity is inhibited, once again, with the addition of anti-tissue factor antibodies. In 2022, the same group investigated the clotting potential of different body fluids. So together with milk and saliva, they also investigated amniotic fluid and urine. When they added this fluid to normal plasma, they could show that indeed, so as you can see from the short bars, they induce clotting, and to a different extent. So for example, milk is the most potent between them, followed by amniotic fluid. Now, when they added the same fluids to factor 7 deficient plasma, they show that still the fluids were able to induce clotting. And this clotting was completely abolished after incubation with anti-factor 7 antibodies. They performed size exclusion chromatography. They have taken the EV fractions of these fluids, and they have they have measured the factor 10A generation with a chromogenic assay in which there is no adenine from plasma. And they have shown indeed that these fractions are able to generate factor 10A, that again, this is abolished, completely inhibited by anti-factor 7 antibodies, and it's strongly dependent on the factor 10. So what is here is shown is that in the body fluid, in those body fluids, we have all of the three components that trigger clot formation. We have the vesicle, we have the tissue factor, and it's complex already to the activated factor 7. What I didn't mention is that tissue factor and factor 7A form a complex which is called the extrinsic tenase complex. In the plasma, where is no factor 7, nevertheless we have the substrate for the tenase complex, which is activated and then lead, as we have seen, to the formation of fibrin, of clots. Why would this be of interest? We have an example. So hemophilia is a blood is a condition in which persons lack either factor 8 in hemophilia A or factor 9 in hemophilia B. And it is shown that the observation is that in persons with hemophilia, oromucosal bleedings are rare, and the hallmark of hemophilia are joint bleedings. Now, I have not talked about factor 8 or 9 so far. I will do it in a moment. If we compare this picture to persons that lack factor 7, we see something different. We see that mucocutaneous hemorrhages are frequent and serious. And as one of the main experts of coagulation factor deficiency wrote, that is Flora Peivande, the reasons for this behavior are unknown at the moment. It was 1997. So I told you that the main trigger of coagulation is the complex formed by tissue factor and factor 7A that generate factor 10A. But this generation of factor 10A is amplified by a complex formed by activated factor 9 and activated factor 8. Now, in hemophilia patients, there is not such a amplification because the complex is simply not there. So there are relatively small amounts of factor 10A generated in thrombin and fibrin. So what we suggest is that additional tissue factor that is provided by the vesicle present in saliva compensate the lack of factor 10A generation, which should have been done by the factor 9, factor 8 complex. And since there is a lot of this complex, this tissue factor, factor 7A, there is a good, a strong and a fast clot formation. We have in our study collected saliva from persons with hemophilia A, so the blood factor 8, and we added these salivas to normal plasma and to factor 8 deficient plasma. And what we have seen is that this saliva is able to shorten the clotting time equally in both plasmas. So meaning that factor 8 is not needed. So, and we have then collected saliva from factor 7 deficient persons. We added to factor 7 deficient plasma and we observed no clot formation. And of course, persons that lack factor 7 in blood lack also factor 7 in saliva. So in saliva, we have not the full complex. So in our retrospective analysis of rates and sites of bleeding in persons with hemophilia A, we have seen that oropharyngeal bleedings stopped spontaneously and joint and muscle bleedings needed administration of factor 8. So we conclude this study saying that the presence of these vesicles, which are also present normally in physiological situation, in case of factor 8 deficient persons, gives protection towards mucosal bleeding, which is not the case in factor 7 deficient persons. So meaning the presence of these vesicles may have an impact on the bleeding phenotype.
Thank you, Carla. I take now over, just share my presentation. Okay, thanks, Carla, for showing our research. And actually, we were quite intrigued by these findings and really believed, particularly in our experiments with milk, that this is a completely new observation. So, of course, what I did when we found first that the milk is so coagulant, literature research via PubMed in English, and there was no finding of any publication showing that there are these tissue factor or factor 7 complex exposing UVs. But then I was wondering whether we searched thoroughly enough in the literature and whether we went back in time long enough, really, if there are really no previous publications on the coagulant potential. And then what we found was quite surprising, namely that there is plenty of really old data on the coagulant potential of body fluids. And interestingly, it has to be said that this data is still very valid because the coagulation diagnostics did not change so much in the last 100 years in large parts. And I will show you what we found. And this just evolved, so to say, parallel to our publications, or even afterwards, we found that there are these intriguing old publications. And I wrote a century of evidence, as indeed the first paper on the coagulant potential of saliva dates back to 1928, and that's at least the oldest paper I found. And this was really quite simple but intriguing. So the surgeon John B. Hunter from the Royal Northern Hospital in London, he simply measured, he mixed saliva 50% with 50% full blood, And then what he found was that there was a shortening of clotting time. So if he used saline, blood clotted after 15 minutes, and with saliva after 2 minutes. So he concluded that saliva is coagulant, and this was the first publications. And research went on. About 10 years later, Alphonse Solé, a Viennese pediatrician, he found out that mother's milk is highly coagulant. And why did he do that? He was treating boys with hemophilia, and at this time there was absolutely no treatment of hemophilia. The life expectancy was extremely short of these boys. They died frequently at young age. They had severe problems, were immobile, could not move because of the joint bleedings and so on. So he was desperately looking for a treatment, and he had somehow the idea to mix mother's milk, he said, because it can clot with the other fluid that can clot with blood. And he thought this affected it, induces immediate clotting. And what he then did, he didn't only describe this phenomenon, which is not present in cow milk. He also performed what we call today a clinical trial. So he applied in a very standardized way tamponades soaked with human milk to external bleedings of boys. And this really stopped effectively the bleedings, and it was, so to say, the first effective treatment of hemophilia. This publication is only available in German, and that's why I didn't find it when we started with our trial. But we now also translated it the main findings into English, and this is in the publication and the review we have published recently. So this was also even in the media. I found a newspaper report in the Spanish newspaper Diario de Madrid, and they say it's like the cure of hemophilia. This is a bit of an exaggeration, but indeed it was the first treatment of bleedings in boys with hemophilia. Research went on. Then again in 1957, Noor Eldin and Wilkinson investigated the coagulant potential of saliva again, and then they did something interesting. They just mixed saliva with different kind of plasma, and they used factor deficient plasma. So these are plasma from hemophiliacs at this time. So, and what they found is quite comparable to what we found, namely that, of course, normal blood saliva is highly coagulant, but also in hemophilia blood, which is factor 8 deficient blood and factor 9 deficient blood. And surprisingly, it must have been surprisingly at this time too, in factor 7 deficient plasma. And that's exactly what we found. And now with our current knowledge, we can conclude this is also in these investigations due to the presence of extracellular vesicles that expose tissue factor and factor 7A complex together called the extrinsic tenase complex. Research went on. Also, urine was investigated. Here they showed that the coagulant potential after ultracentrifugation is in the urine pellet and not in the supernatant, pointing again to the presence of vesicles. And also they performed filtering experiments, so putting the urine through a 0.2 micrometer filter removes the coagulant potential of the urine. In this very nice study by Wiggins et al. A bit later, 87, they again used this factor deficient factor 7 deficient plasma and added urine, and they already concluded that this procoagulant activity must be factor 7-like, and they already had the right conclusion that these findings are compatible with the concept that particles carrying procoagulant activity comprise thromboplastin with factor 7A. So to say, exactly the same what we concluded from our research a long time later. They also performed very nice electron microscopy with rabbits, and they had glomerular nephritis. And here you see the blabbing on vesicles from this glomeruli. And why they investigated this? Because it's a typical pathologic picture that when there's glomerulonephritis, that you have these casts in the nephron, which are basically fibrin depositions. So they were following this path because it's a well-known, so to say, pathognomonic picture of tissue damage in the glomerulum. Another group from Scripps, also a bit later, 97, investigated the procoagulant potential of semen. And this is interesting because prostosomes is one of the earliest terms for extracellular vesicles. So it's physiological for prostate to secrete vesicles. And they found this coagulant potential to be extremely strong. So they diluted it 1 to 10,000, and even at this dilution, semen was highly coagulant in plasma. And again, they used factor 7 deficient plasma. And this time, for the first time, they also used factor 7 monoclonal antibody. And here you see that also in the factor 7 deficient plasma, semen is coagulant, and this is completely blocked by factor 7 antibody. And they also performed differential centrifugation and saw that the coagulant potential is only in the pellet. And it is really in the prostatic fluid as they collected it during prostatectomies. So I think evidence from this study is so strong that we can really say and confidently that these are tissue factor 7A complex exposing EVs. This paper, as also the other papers, seems to have been completely forgotten. They also performed electron microscopy like Rienk did in his study, and also here they could show with gold-labeled tissue factor beads that these vesicles expose tissue factor. With amniotic fluid, a quite similar experiment was performed. It is well known that embolization of amniotic fluid in the maternal circulation during delivery induces DSC, and therefore there was quite a lot of research on the coagulant potential of amniotic fluid. And again, amniotic fluid is also coagulant in factor 7 deficient blood. Another group showed that all this coagulant potential is membrane bound and palatable, so associated with vesicles. And again, we have this odd conclusion that there must be tissue factor 7A complex exposing vesicles in amniotic fluid. So in our review, we looked at all kinds of physiological body fluids, and what we found is that amniotic fluid, milk, saliva, semen, and urine clearly have coagulant potential that depends on this tissue factor 7A exposing extracellular vesicles. Also, tears are highly coagulant, and most probably they also have these vesicles have also factor 7 on the surface. However, only tissue factor was investigated, so there still needs to be the proper study performed, but most probably also tears have these vesicles. So what are the conclusions from this? EVs exposing functional extrinsic kinase complexes, so tissue factor and activated factor 7, are present in many physiological body fluids. And these are some kind of hemostatic agents that every one of us carries, or most of us carry, and they protect from bleedings, and they have also an impact on the bleeding phenotype. So either if we hurt ourselves and lick our wounds, but also in persons, for example, with hemophilia A, who don't have these typical mucosal bleedings, in contrast to persons with factor 7 deficiency who have these mucosal bleedings. So these are observations we further follow. And we would like to thank our contributors. This is primarily a cooperation between Medical University of Vienna and with Rienk Nieuwland and his group. It's really a joy going on for now several years. Ideas going back and forwards. We help each other with experiments. And I think we have some nice publications out of this collaboration. Particularly, we would also like to thank Wolfram Ruf from the Johannes Gutenberg University in Mainz and Scripps, who provides this very nice monoclonal antibodies for the different coagulation factors we investigate, which really helps us to find out what is going on. And we want to thank Alain Brissot from the University of Bordeaux for these very nice electron microscopy pictures he made for us. Yeah, now it's the presentation is open for discussion.
Great. Well, thanks very much to all three of you for that excellent presentation. I always enjoy hearing about some of the history too, putting everything into context. That's really really educational. So for those of you who are not presenters, I have now enabled you to unmute yourselves and to show your cameras if you'd like to. But please put your questions in the chat box so that we can be sure to get to you and call on you. I would just like to start. So given, I think there's been a question in the EV field, and especially in the liquid biopsy corner of the field about tissue EVs and how common it is for them to get into biofluids like the blood. And there's been a very wide range of estimates. So for example, I work on the brain, and the estimates range from something from just a tiny fraction of 1% of EVs in the circulation being from the brain to as high as higher than even 10%. Some folks have said, and of course the brain is not the only organ in the body by any means. So how much is too much? When does it become a problem? At what level of EVs coming from tissue do you start having problems with their contribution to coagulation, or is it not just a matter of numbers? I suppose it's also a matter of what exactly is on the surface.
I think I can answer part of it because it was part of my PhD thesis actually. Because I started with investigation of tissue factor, now I believe tissue factor 7A complex exposing Vs in blood of cancer patients. And here it seems to be a big problem early because it's just not meant to be there circulating free in blood. And what I found was that these vesicles that are coagulant are primarily present in patients with advanced disease. And in one study, we investigated levels of these vesicles in pancreatic cancer patients, and pancreatic cancer is full of tissue factor. And we observed that the high levels were only present in those patients who had also tumors that infiltrate large abdominal vessels. So it seemed really that the tumors needed to have access to the vascular system to vesiculate directly. There's another type of leukemia. It's called acute promyelocytic leukemia, and in this leukemia, the blast secrete tissue factor exposing vesicles, which is kind of part of the genetic program, and but it's very quickly. It's also low levels are too much. It causes trouble and needs to be treated immediately. Otherwise, the patients usually die within a short period of time. And it's very different. It seems to be extremely different when you look at the body fluids, which are in contact with epithelia. So there is a completely different effect. And for example, looking at mother milk, you would regard it as active as a snake's venom, right? You would think it's poisonous. For sure, it would be dangerous intravenously, but it does the baby well, right? It's the best food for them. Still, I would not have imagined that it is that coagulant. That's my. Rienk, you have something you wanted also to?
Yeah, so Ken, I think also because it's a good question, but so if you assume that the vesicle from a tissue is exposing tissue factor and perhaps factor 7, or it activates factor 7, there's also the natural system of checks and balances in the blood, and we have tissue factor pathway inhibitor. So you need to go over a certain threshold before I think tissue factor will really be able to trigger clotting. So there is it cannot be in such a way fine-tuned that if you only get one molecule of tissue factor in the blood, that you will die from venous thrombosis. So there's also the natural inhibitors of the blood clotting around. The problem is, in a very old paper, we showed, for example, that if you isolate vesicles from wound blood from patients undergoing open heart surgery, and you inject them in vein of a rat, it will almost immediately develop a thrombus because it's highly coagulant. And if you put in vesicles about the same quantity from healthy human individuals, nothing happens. So it's really tissue factor 7 there that apparently there's such a bulk that it triggers clotting. But not every little vesicle will make you die. I mean, it's not that bad.
Yeah, I think I'm thinking of this from the perspective of some of the folks who would like to open up some barriers between the blood and tissues, you know. So whether that's to I'll give a specific example. Let's say that we can open up the blood-brain barrier slightly, maybe using focused ultrasound, and maybe using something to change something about the property of the blood locally. The idea there is we would be able to have more of an outflux and efflux of EVs from the brain, but perhaps also have an opportunity to put therapeutic EVs into the brain. Now that efflux is what I'm thinking about, you know. So because if indeed we could be causing a problem with the efflux, maybe we don't know about a cancer that's present, or, you know, so there might have to be a little bit more thought that goes into those sorts of proposed interventions before we do them based upon your findings. Is that, do you agree with that, or am I being too much of a scaremonger?
Well, I just have to think that actually the very first Western blot I ever did for tissue factor, the positive control were minced human brains, because they're really full of tissue factor. I mean, it's the ultimate positive control that was being used in the labs. And so, yeah, if you think that an efflux of vesicles from the brain may come into the peripheral blood and they are exposing tissue factor, it doesn't seem healthy to me. But Johannes, what do you think, or Carla? I mean,
to my knowledge, cerebrospinal fluid is not coagulant significantly, for example. But so in this regard, I would not expect that it induces clotting. Interestingly, for example, I know another example is a pathologic fluid, namely ascites, from patients with liver cirrhosis. And what they did more in the 80s, they had a shunt from the ascites into the blood circulation. So so ascites is water, or a liquid from a transsudat, so some fluid in the abdomen, and it is directly infused into the blood circulation. And what they observed then was indeed that the persons developed DIC afterwards. And we know that ascites contains tissue factor, likely tissue factor 7A complex exposing EVs. So this can induce DIC. And amniotic fluid embolism, which is a very rare complication of delivery, which was likely much more frequent in previous times. On the one hand, it induces shock, but also typical sign is disseminated intravascular coagulation. So I think this is something you have to consider, that this hemostatic system is good to stop external bleedings, but it's so effective that it would cause trouble when it's in the intravascular system.
There's a reason why we have barriers in the body, right?
Exactly. And also the blood-brain barriers has a reason to prevent infections, right? So this would be what I would be more concerned of. There's no way, you don't want any germ, right, in your central nervous system. That I would be more concerned of if you want to disrupt the blood-brain barrier, actually.
But I'm actually impressed to hear that Rienk, your first Western blot worked. Is that right?
It worked, of course, yeah.
I think most of us cannot say that, but
it was thanks to the positive control. I mean, that's sort of
very good. Yeah. All right, well, we have a question in the chat box from Camille. Camille, could you tell us what you're thinking?
Hi, thank you very much. I don't know, can you hear me?
Yes, we can.
Thank you. Excellent presentation, all of you. Thank you very much for the presentation. I have a question about TFEV in sweat. I know Rienk has done some work already on this, but it's not present. There is different kinds of sweat. There is sweat which we use for cooling down, and sweat when we are frightened, or we are about to be eaten by a lion, or we are about to fight somebody with a sword. Now there's fright and there is cooling down. Cooling down sweat does not have TF activity. But if you're frightened, it's a different matter. This, to me, means that your body is preparing itself for an impending bleeding. And this also has an implication on wound healing in future. I would like some comment from, please.
Yeah, well, I I have to laugh because my dream is still that I would like to do a sort of similar experiment for tears, collect tears from people who are really afraid and people who are really emotional, and then study how much tissue factor activity is present in those tears, because sweat is pretty difficult to collect, to be honest, but tears are a bit easier. And let's say we have PhD students giving the first international lecture somewhere over the world and collect their tears when they're crying because they're afraid, or tears when you look at a scary movie or whatever. And yeah, I think that would be that I would not be surprised that there are really differences there in body fluids like sweat and tears regarding the amount of coagulant tissue factor and 7 that is there. Yeah, for sure. But it's a nice idea, but it's not so easy to perform. So if anybody has a good idea, I would be, or would have samples, I would immediately measure them.
Go to a kindergarten is maybe the best.
Or so.
But the ethical committee may not.
Yeah, there may be some medical ethical problem with these samples.
I don't know, maybe some people who are about to, jump off a platform and do some bungee jumping or something like that.
Exactly. Oh yeah, exactly. Yeah. So
They're
things like
doing it
that.
voluntarily already, so they can sign up.
Yeah. Reenacting D-Day. or
Rienk,
something.
you cut onions, right? You let students cut onions, I think, right?
Yeah, we did a few training for medical students. We had some of the medical students had to watch Bambi or another sad movie, and the others had to cut onions. And we collected tears and looked at the tissue factor, etc. But it's not so black and white in the end. There's a lot of tissue factor there in clotting activity, but you cannot really see different levels.
May I ask another question?
Please go ahead.
This time is probably to Johannes. Tissue factor is very high in brain, as already has been mentioned. It's also localizes where with the brain of neurodegenerative disease patients, including Alzheimer's, is present in the brain fluid in Parkinson's disease and other neurodegenerative disease. Is it playing a role there? I would like very much your opinion.
I have honestly to say, I don't know if it plays a role in the pathogenesis of degenerative diseases, but for sure it plays a role in the physiology of the brain, namely protecting from intracerebral bleedings. Because I looked a bit at patient histories of persons with factor 7 deficiency, and seizures, epilepsy is frequent in these persons. And I found it quite interesting because if a toddler learns to walk, right, they fall down and then they cry heavily, and we just say, oh yeah, that's normal, they're crying, they're babies, they're crying, haha. But it seems that they really suffer normally quite severe brain damage. But the brain is full of tissue factor. They have factor 7 in the blood, so this stops the bleeding. And I know patients with factor 7 deficiency who had to have neurosurgery because of scars in the brain from old bleedings. So also this is a hemostatic mechanism in the brain, which is really important, it seems. Yeah, but I don't know, I have to say, how tissue factor and in some pathologic other conditions contributes.
Thank you for your opinion.
Thanks for the questions. I see that the president of the Austrian Society for EVs has his hand up. Wolf?
Yeah, thank you very much, Carla, Johannes, and Rienk, for your presentation. Very interesting, this issue with the tissue factor on the EVs. I'm just thinking in terms of therapy or therapeutic applications. So if you have EVs and they carry a tissue factor on the surface, you might run into a problem with coagulopathy, right? And so what are your thoughts about this? Because we are now trying to CRISPR out the tissue factor from EVs in order to delete this problem. So do you think that EVs which carry tissue factor might be suitable for therapeutic applications in several diseases? I think of cardiovascular disease. I mean, you would not want to receive EVs with tissue factor which clot your blood, right?
Can I comment? Because I'm as an MD, I thought about it a lot, and absolutely not. I think that's the wrong. So these vesicles only do harm intravenously. There's already a drug you for sure know. It's activated factor 7, Novo7 on its own, and it has a completely different mode of function. It's just 200-fold supraphysiological activated factor 7 at the therapeutic concentration. And what makes this quite ingenious is that it's at this concentration, it can bind negatively charged phospholipids and activate coagulation locally at the site of injury. And therefore, it's an emergency drug, also on the battlefield or after trauma and so on, to stop bleedings. Tissue factor 7, exposing vesicles intravenously would only induce TSC. It could work as a topical hemostat, as Alphonse Solé did in 1934. There it would be very effective. But I think there are all, and you know it better, I think, there are several very effective hemostats already, tamponates and so on, with activators of coagulation on the market. So I think the way to go for vesicles without tissue factor is, seems way better. Yeah. So I agree. Thank you.
Okay, thank you very much, Johannes, and also for your great presentation.
Thank you.
Thank you, Wolf. Do we have any more questions from the audience today? Then if not, I would just like to thank you, Johannes Thaler, Carla Tripisciano, and Rienk Nieuwland for your presentation, and of course for the lots and lots of work that went into this. I know that you're continuing this work, and we wish you all the best as you do that. And we look forward to hearing more from you. So thanks everybody for being here today, and we hope to see you on a future EV Club sometime soon. So thank you, and have a great rest of your day, everyone. See you soon.
Thank you.
Thank you very much.
Thank you.
Bye-bye.